Proteins are one of the 4 major macromolecules of life. Macro meaning they're big molecules and you need a lot of them, all right, in order to survive, along with carbohydrates, lipids, and nucleic acids. But proteins serve so many important functions in your body. You probably know these already. So first of all, obviously, proteins provide support. Muscles, right? They're made up of proteins. Your cells are made up of proteins. Everything in your body consists of proteins. They're really important for your metabolism. All enzymes in your body are made up of proteins, and enzymes catalyze all the reactions in our body. They're important for transport, so moving things around in the body, moving things in and out of cells. Defense, your entire immune system consists of these proteins, so important. Regulation, so turning on and off gene expression, and homeostasis, maintaining your body's stability. And last but not least, motion. All movement requires proteins. So proteins are essential. Proteins are made up of components, monomers, right? The building blocks of proteins are known as amino acids. There are 20 different amino acids, and they all consist of the same basic structure, and this is what the structure looks like. So the center of the amino acid is always a carbon. Attached to the carbon is a single hydrogen. All amino acids have these things, okay? They all have a carbon, hydrogen, a carboxyl group, which looks like this, C bonded to OH, double bond to oxygen. And on the other end is the amino group. Amino group, I always remember N in amino stands for nitrogen. So it has a nitrogen and 2 hydrogens. The only thing that makes every amino acid different is this last group called the R group. So the R group, this what gives the amino acid its properties, whether it's going to be acidic, basic, hydrophilic, hydrophobic, all determined by the R group of the amino acid. So here is a chart of all 20 amino acids and their structures. And obviously don't need to memorize this for this class, but what you just want to look at are how the R groups, okay, pictured here in orange, blue, pink, or green, how they're all just slightly different, and that's what gives the amino acid its properties, whether it's acidic or basic, polar, indicating that it likes water. These amino acids are attracted to water and they will move with it with the water. Or the non-polar amino acids in this group, meaning they do not like water, they're repelled by water, they stay away from it. This group here at the bottom, these are kind of the weirdos. Some of them have like sulfur in them or these weird aromatic ring structures. So every amino acid has different characteristics distinguished by the R group. Now the way amino acids, they come together and they form a special type of bond called a peptide bond. This is a very strong bond. It's actually a type of covalent bond, very strong. And what happens is the carboxyl end, the end with that carbon, of one amino acid hooks up with the amino end of the other amino acid. And what happens is this OH and this H get chopped off. So water, H2O, 2 H's and an O, gets released. And then these two amino acids can hook up and they link together, forming a peptide bond. So after this reaction, it's actually called a dehydration reaction because you're losing that water. When you lose water, you become dehydrated. You know that. So we now have these 2 amino acids hooked up and we lost that water. And then this happens many times. So you get many amino acids that hook up to each other and they form this long polypeptide. And that's what the building block of a protein looks like. Remember before when I was showing you the structure of amino acids and I said there's one end that's the amino group and one end that's a carboxyl group. So the amino group has the nitrogen and the carboxyl group has that carbon
with the O and the OH? Well, naturally, when a bunch of amino acids are going to come together, one end is going to have that exposed amino group, so that nitrogen at the end. And the other end is going to have that exposed carboxyl group with the carbon at the end. So somebody who was like, let's come up with a name for this, they came up with a really easy name. The end that has that free nitrogen is called the N-terminus, and the end of the amino acid sequence with the carboxyl group is called the C-terminus. Don't forget to look over the review questions. Now, when a protein is made, there's 4 levels of organization of the protein. The first level is called the primary structure. The primary structure is simply the sequence of amino acids in a chain. Okay, so just like I showed you before, it's going to be a chain of amino acids with one end having the amino group, the N-terminus, and somewhere at the other end, you should find the C-terminus. So that's the primary structure, simply just the sequence in order. The secondary structure, in the secondary structure, that chain of amino acids is going to fold. And there's 2 different types of folds that could happen. One of them is called an alpha helix. It kind of looks like a ribbon. This is what it looks like. You see this chain? That would be an amino acid sequence that forms this alpha helix. Or the second type of fold is called a pleated sheet. You know when you were a kid and you used to make those paper fans and you would fold the paper in and out? That's what a pleated sheet looks like. So that's the other type of fold the amino acids could do. The tertiary structure is when a bunch of alpha helix and pleated sheets come together and they form a glob. So when you see a big globular structure that contains some of these alpha helix and some of these beta sheets that are all together, that's the tertiary structure. Finally, the quaternary structure is when multiple tertiaries come together. So you get some of these blue globs, some of these red, some of these green. And the quaternary structure, this is the final protein that hopefully will be able to act and perform its function. Now, a couple of things could happen that could ruin the function of proteins. One of those things is the protein could become denatured. This is basically a fancy word that means to melt. So the protein can basically melt. And when the protein melts, it loses its structure and then it can't function properly. There's 2 ways a protein could denature. It can either be due to extreme pH, such as an acid, or temperature, like really hot temperature. So for example, if us humans were to jump into a volcano, all of our proteins would melt and denature because of the heat. That's kind of what happens to an egg. When you fry an egg, the proteins will become denatured. It'll go from a normal egg to a denatured egg. And once a protein is denatured, it could never return back to its normal state. That's why you could never crack an egg and then try to put it back. It wouldn't work. Proteins are denatured. Now here's another thing that could happen. You might have heard of these things called prions before. These are infectious proteins. The way somebody would get a prion disease is from eating contaminated meat that contains these infectious proteins. So here's what prions do. Inside of our brains, we have these normal prion proteins that are shaped like this with alpha helices, right? And these proteins act normal and you're totally fine. But if somebody gets exposed to this infectious prion protein, this protein changes in shape. So you see these red alpha helices, they become pleated sheets. So this protein changes its structure and it doesn't function properly anymore. And what can happen over a period of time is these infectious proteins can accumulate in the brain and they can form these holes, like spongy holes in the brain, and this leads to dementia. You might have heard of one of these diseases called mad cow disease, which comes from eating cattle that's infected with the prion protein. So these are examples of prions, misfolded proteins. The last thing I want to mention about proteins is sometimes somebody could have a genetic change in the normal sequence of proteins, and this could lead to a genetic disorder. And the one example we're going to talk about 10 times this semester because it's a really good representation of this is sickle cell disease, also known as sickle cell anemia. So here's what happens with someone who has sickle cell anemia. Here is a normal hemoglobin. This is a red blood cell. Hemoglobin is a red blood cell. Now normally red blood cells are nice and round so they can fit through blood vessels and they can carry oxygen efficiently, and they have almost like this little donut kind of shape. They're little concave in here. And the reason that these red blood cells are shaped like this is because the proteins fold in that way. So the protein sequence allows the red blood cells to fold properly. In sickle cell anemia, an individual inherits one single mutation in the amino acid sequence. So the primary structure of the protein has one change. This glutamate has been mutated to a valine. And that one single small itty-bitty change is enough to change the entire structure of the red blood cell. So instead of the red blood cell being nice and round, it develops a sickle shape, which looks like this, kind of looks like a banana or like a crescent moon. All right, so that's what sickle cell anemia is. It's caused by this one amino acid change. And because the red blood cells are shaped like this, they cannot fully carry oxygen as well. And also they could get clumped up and lodged into the blood vessels. So that was it for proteins.